Enzymes are the molecular machines that power life. Discover how they work. Catalysis, kinetics, inhibition, and regulation. Enzymes control the chemistry of life. Subscribe now and unlock the secrets of biochemistry with us.
Sucrose oxidation is a highly exergonic reaction. The conversion of table sugar into carbon dioxide and water releases a massive amount of free energy. Yet, a bag of sugar can sit on a shelf for years without reacting. The process is thermodynamically favorable, but on a biological timescale, it is kinetically non-existent. This delay exists because stable molecules must absorb a massive influx of energy to reach a transition state before they can react. This diagram maps free energy against reaction progress. Connecting substrate to products is a massive peak, the activation energy, representing the unstable transition state. But the secondary, lower curve is the enzyme-catalyzed pathway. It collapses the barrier, allowing the reaction to proceed in seconds. Enzymes accelerate the journey to equilibrium, but they operate under a strict thermodynamic limitation. They cannot alter the standard free energy change of the reaction itself. The starting and ending energy states remain exactly the same. To understand biological engineering at its highest level, we must decode exactly how enzymes physically manipulate these microscopic energy barriers. In 1894, Emil Fischer proposed the lock and key hypothesis. He suggested that an enzyme's active site was perfectly structurally complementary to its specific substrate. The flaw in this model is thermodynamic. Perfect structural complementarity stabilizes the substrate in its ground state. A highly stable ground state effectively impedes catalysis. Look at the left side of this split screen. We are modeling an imaginary enzyme, a stickase, using magnets to represent binding energy. Here, the straight metal stick fits perfectly into a magnet-lined pocket, replicating the lock and key model. On the right side, the graph shows the consequence. Because the stick is perfectly stabilized, the enzyme-substrate complex plummets into a deep energy well. The required activation energy to bend and break the stick is now higher than the uncatalyzed reaction. This suggests a different structural requirement for the active site. If the enzyme isn't shaped for the substrate as it currently exists, we have to look at what the substrate becomes during the reaction. Linus Pauling formalized this structural insight in 1946. Applying Pauling's theory to our model, the enzyme pocket is now curved. The straight stick binds weakly at first, but as the stick bends toward the transition state, all the magnetic interactions perfectly align. This alignment releases free energy, known as binding energy. Forming these weak, non-covalent interactions exclusively in the transition state pays for the energetic cost of substrate distortion. On the graph, the negative binding energy is subtracting from the positive activation energy, resulting in a drastically flattened peak. These results show that the active site is an energetic trap. It uses binding energy to force a substrate out of its stable ground state and into the geometry required for a reaction. The core challenge of enzyme kinetics is measurement. The enzyme-substrate complex is fleeting. We cannot easily measure its concentration in real time. Here is the foundational equation for enzymatic catalysis. It breaks down into two steps: the fast reversible formation of the ES complex and the slower breakdown into the product. Each step is governed by specific rate constants. J.B.S. Haldane provided the theoretical contribution needed to bypass the unmeasurable ES complex. This chart plots the macroscopic observable data. We map the initial velocity of the reaction against an increasing substrate concentration. The result is a hyperbolic curve, where the reaction rate plateaus at high concentrations. Along with G.E.
Briggs, Haldane introduced the steady-state assumption. Assume that the rate of ES complex formation is exactly equal to its rate of breakdown. This is the algebraic translation of that assumption, balancing the forward rate constants against the breakdown rate constants. By isolating the ES complex mathematically, unmeasurable variables are substituted with the measurable total enzyme concentration. The separate rate constants bundle together into a single term, the Michaelis constant, or Km. Substitute this back into the initial velocity equation, and we arrive at the final Michaelis-Menten equation. Vmax is the absolute velocity ceiling. It is achieved only when the enzyme is entirely saturated with substrate and cannot process molecules any faster. Km is the exact substrate concentration required to reach 1/2 of that maximum velocity. Km is widely cited as a direct measure of binding affinity. This is incorrect. Because Km bundles multiple rate constants together, it only reflects binding affinity in rare cases where product release is overwhelmingly rate-limiting. The Michaelis-Menten equation provides the framework to convert unmeasurable, fleeting physical interactions into predictive, macroscopic kinetic constants. Biochemists use enzyme inhibitors as structural probes, allowing us to stress test the Michaelis-Menten model by observing how the system responds to specific interference. By taking the double reciprocal of the Michaelis-Menten equation, we transform the saturation curve into a linear Lineweaver-Burke plot. This isolates our discrete parameters. The y-intercept is 1 over Vmax, and the x-intercept is -1 over Km. In competitive inhibition, an inhibitor strictly binds the free enzyme, blocking substrate access. This produces a precise kinetic signature on the graph. A family of lines pivots strictly around the y-intercept. The x-intercept shifts towards 0, representing an increase in apparent Km, but the y-intercept remains anchored. The y-intercept stays anchored because an infinite concentration of substrate will eventually outcompete the inhibitor. The true maximum velocity is entirely untouched. Contrast this with uncompetitive inhibition, where the inhibitor ignores the free enzyme and binds exclusively to the already formed ES complex. The kinetic signature changes completely. The plotted lines shift upward and to the left in perfect parallel. Both intercepts change, indicating a drop in both apparent Vmax and apparent Km. By mathematically mapping how a kinetic system fails under specific perturbations, biochemists deduce the exact physical architecture of an invisible active site. The turnover number, kcat, defines the maximum number of substrate molecules converted to product by a single saturated enzyme per second. The ultimate metric for an enzyme's overall catalytic efficiency is the specificity constant, the ratio of kcat to Km. This chart highlights a highly specialized subset of enzymes. Acetylcholinesterase and carbonic anhydrase achieve staggering specificity constants in the range of 10 to the 8th to 10 to the 9th. Here is what those massive kinetic numbers represent on our original reaction coordinate diagram. In these enzymes, the binding energy has flattened the chemical activation barrier to 0. When the chemical barrier is negated, the only remaining speed limit is the rate at which molecules physically bump into each other in an aqueous environment. This is the diffusion-controlled limit. An enzyme operating at this limit is constrained only by the physics of the environment. At this point, catalysis is no longer the bottleneck. The speed of life is dictated by the time it takes for two molecules to drift together in water.
Within cells, water molecules collide aimlessly with stable protein backbones. Without structural intervention, spontaneous cleavage of a single peptide bond takes hundreds of years. To bypass this energetic stalemate, biology employs precise molecular machines. Bovine pancreatic chymotrypsin, isolated from these tissues, is one of the most efficient solutions studied. The enzyme's architecture consists of 3 distinct polypeptide chains, covalently linked by bisulfide bonds, and folded into a dense, specific tertiary structure. This arrangement accelerates the cleavage of peptide bonds by a factor of 1 billion compared to the same reaction occurring in bulk water. Catalysis begins with recognition. A deep hydrophobic pocket on the enzyme surface is sized to anchor bulky aromatic residues like phenylalanine and tryptophan. Positioned precisely at the edge of this pocket are 3 residues: serine 195, histidine 57, and aspartate 102, which coordinate the actual chemistry. By combining pre-steady-state kinetic data with structural analysis, we can deduce the exact atomic movements that drive this acceleration. Understanding this billion-fold rate enhancement requires us to deconstruct the sub-second transitions of these residues. In 1954, researchers B.S. Hartley and B.A. Kilby used the artificial ester p-nitrophenyl acetate to investigate how chymotrypsin processes its substrates. Cleaving this ester releases p-nitrophenyl, a molecule that turns the solution yellow, providing a visible indicator of the reaction's progress. Measurements revealed a biphasic progression, an initial vertical surge followed by a slow linear rate. This burst phase is stoichiometric, releasing exactly 1 p-nitrophenyl molecule per enzyme instantly. The reaction then enters the steady state,
where releasing a second product becomes rate-limiting. This suggests a two-phase mechanism, a rapid acylation that primes the enzyme and a slow deacylation that resets it for the next substrate. This kinetic burst is the signature of a transient covalent intermediate, a physical chemical bond formed between the protein and its substrate. To identify which residues manage the protons during this sequence, researchers track how changes in pH affect the catalytic rate. Plotting reaction velocity against pH generates a bell-shaped profile with a clear maximum at pH 8.0. Below pH 7, activity drops because histidine 57 accepts a proton. To function, this residue must remain unprotonated so it can act as a general base. Above pH 9, activity falls again. At this alkalinity, the alpha amino group of isoleucine 16 loses its proton, destroying the salt bridge that stabilizes the active conformation of the binding pocket. Within the active site, aspartate 102 shortens the distance of its hydrogen bond with histidine 57. This creates a low-barrier hydrogen bond that shifts histidine's pKa toward 12. This electrostatic pressure allows histidine to strip a proton from serine 195, transforming it into a highly reactive nucleophile. The activated serine oxygen attacks the substrate's carbonyl carbon, forcing the planar peptide bond into a tetrahedral geometry. This attack generates a localized negative charge on the carbonyl oxygen, an extremely unstable high-energy state. This charge is stabilized within the oxyanion hole, where backbone amide nitrogens from glycine 193 and serine 195 provide specific hydrogen bonds. These stabilizing bonds are not present when the substrate is in its ground state. They only engage once the molecule begins to deform. This selective electrostatic support significantly lowers the activation energy, enabling the billion-fold increase in reaction speed. The tetrahedral intermediate then collapses, breaking the peptide bond and releasing the first product fragment. The structural evidence points to an active site configured specifically for this short-lived geometry rather than the stable substrate. To verify the enzyme binds the transition state more effectively than the ground state substrate, we examine synthetic analogs. By adding extra chemical groups far from the cleavage site, we measure how peripheral interactions affect the reaction. This table compares the modified substrates. The binding constant, Km, remains relatively unchanged, while the turnover number, Kcat, increases exponentially. These extra groups barely affect initial binding, but provide a massive boost to the speed of the reaction. Because these groups only find their binding partners during the transition state, they prove that the active site functions as a negative mold of the reaction's high-energy barrier. A mechanism this powerful is a biological hazard. An active protease is a chemical weapon that will dismantle any protein it encounters. If chymotrypsin were active during its synthesis, it would cause indiscriminate damage to the internal environment of the cell. To manage this, the enzyme is synthesized as an inactive zymogen called chymotrypsinogen. In this state, the active site is distorted and non-functional. It only reaches its billion-fold potential after an irreversible proteolytic cleavage in the intestine snaps the catalytic triad into its final, lethal alignment. In the architecture of life, extreme catalytic power must be balanced by absolute regulatory control.
Thanks for joining us. Don't forget to subscribe and get ready for Chapter 7, Carbohydrates and Glycobiology. See you there!